Hey all, so in this video, we're going to be looking at photosynthesis, which is C1.3. We want to be thinking about the interaction and interdependence of photosynthesis. And so how do different structures inside cells create energy from light? And additionally, how are they connected to different factors in the environment? We're going to be looking at page 384 to 394, and the questions we want to be thinking about are how is energy from sunlight absorbed and used in photosynthesis, and then also how do abiotic factors interact with photosynthesis. So photosynthesis is the process by which carbon dioxide is used to predominantly make glucose, but then also proteins, lipids, nucleic acids. And if we're thinking about eukaryotic organisms, that's going to be happening in the chloroplast. And there's two reactions happening in the chloroplast, the light dependent and then the light independent reactions. And ATP is going to be created using sunlight. So that's the light dependent reaction. And then that ATP is going to be used to make the glucose from the carbon dioxide, and that does not require light. And so that would be the light independent reaction. Glucose can be used also by mitochondria in plant cells. So for example, in the roots, yes, plants have both chloroplasts and mitochondria, and the mitochondria can then take that glucose to make ATP for cellular function. Glucose can be stored as starch or used to make cellulose. And so usually it's stored as starch in the roots or in seeds, for example. And so that would be a good example of when mitochondria would be using that starch broken down into glucose. So here we can see the reaction of photosynthesis. This is just the backwards reaction of cellular respiration. So we're taking carbon dioxide and water, and then in the presence of sunlight, that's what these little yellow lines are here, they're going to produce glucose and oxygen. And when you're looking here at this image, this is photolysis of light. So what's going on here is water is needed to make ATP through the process of photosynthesis. And so the light hitting the water breaks it up into its constituent parts. And this hydrogen ion is able to generate a concentration gradient. And then that's the process by which ATP can be made. So when we're thinking about photosynthesis was not always the same in cells throughout history, and the early Earth didn't look the same either. The first cells on Earth would have been archaea and prokaryotes, and they started to photosynthesize without chloroplasts, but about 3.5 billion years ago. And so what that ended up doing is causing oxygen levels to rise on the planet because they were producing oxygen as a waste product from that photosynthesis process. And around 2.2 billion years ago, that cooled down the planet. And so that's called the Great Oxidation Event, and the planet went into a big ice age. This process of having oxygen, a lot of oxygen in the atmosphere, caused iron in the oceans to become oxidized, rusting, and it fell to the seafloor, and then it caused these rust-colored bands in the rocks and the iron that we harvest today. So this is how we know that oxygen changed certain aspects of our planet, and then also how we're able to measure that. With these prokaryotes initially 3.5 billion years ago, they started to photosynthesize just using pigmentations, but eventually through a process that HL students will learn more about called endosymbiosis, prokaryotes were absorbed by other prokaryotes, and slowly over time, those prokaryotes turned into chloroplasts and mitochondria. And we know this because chloroplasts and mitochondria have their own DNA, and it looks like prokaryotic DNA. And they also have their own ribosomes, and it looks like prokaryotic ribosomes. So those are structurally different than eukaryotic organisms. So when chloroplasts or pigments are absorbing light to make ATP, the wavelength of the light available can have a dramatic role can play a big role on what amount of photosynthesis is able to happen. So obviously sunlight is visible spectrum light. And if we're looking here, we can see what is going on with action spectrum. This is the action spectrum is how much photosynthesis is happening, the rate of photosynthesis. And then if we're looking at the absorption spectrum, that's this blue, green, and red line down here, that's how much light is being absorbed. So we can see that light is being absorbed in the wavelengths of 400 to 500 nanometers, and then also between 600 and 700 nanometers. So in the electromagnetic spectrum, short wavelengths have more energy. And so the pigments that we usually see associated with photosynthesis, chlorophyll A, chlorophyll B, beta carotene, these are going to be absorbing more in this short wavelength light. And so this is actually purple blue light here that's being absorbed. And then up here is red light. Green light is actually reflected, which is why most plants look green. If you were seeing a plant that looked white, that would be reflecting all light. So that would be highly ineffective from a photosynthesis perspective. If you saw a black plant, it would be absorbing all light. So it would maybe actually have problems with water retention and overheating. So typically the pigments that we are seeing from plants, you can see purple plants and red plants and stuff like that too. But most plants are green. And what's going on there is there's these three pigments absorbing in this wavelength here. So photosynthesis can be affected by a number of different factors. The three main ones are light intensity, carbon dioxide concentration, and temperature. And so you can see the rate of photosynthesis is changing similarly in these first two, but then differently in the last one. So as we increase the amount of light, the amount of photosynthesis is going to increase until a set point when all the chloroplasts are working, and then it's going to plateau. So no more photosynthesis. It can't be sped up any further than that. Additionally, same thing with carbon dioxide concentration. There will be more photosynthesis until all of the chloroplasts are working. And then at that point, it will plateau. With temperature, what's going on is that the reactions are, the molecules are moving faster. So they're bonking into each other more, which makes the enzyme that makes photosynthesis happen. It speeds that reaction up. But as we've learned about enzymes, if the temperature gets too high, then the enzyme will denature and then it will just stop working. So Rubisco is that enzyme. It's the most important enzyme on the planet because it provides all of our oxygen and allows all of our plants to live. If we get plants too hot, then Rubisco will denature and then it will stop working and then photosynthesis will just stop. All right, so to summarize here, chloroplasts use sunlight and carbon dioxide and water to make glucose, lipids, proteins, nucleic acids, and oxygen, and glucose can be stored as polysaccharides like cellulose or starch. And we can see here this condensation reaction would be glucose molecules reacting to create a long chain of starch molecules, for example, but in that case, it's maltose. Green leaf pigments absorb energy better in the blue, purple, and red range of the visible spectrum. More light and carbon dioxide is going to result in faster photosynthesis up to a point, and then it will stabilize. Higher temperatures will speed up photosynthesis until the enzymes denature. And Rubisco is the most important enzyme on the planet because it allows photosynthesis to happen. Okay, thank you for watching this. That's all for this one.